Yeah, thanks for sharing, everybody. We're going to go ahead and get started. So welcome to our Friday Phenology check-in, our lecture today. I'm Denise Ellsworth, and I want to welcome everybody here. So just a couple notes of housekeeping. I'm going to ask you to go ahead and turn off your video for now. When we do small groups at the end, you can turn your videos back on, see who's there, and share. Make sure you're muted as well. Let's see, Pat and Kathy, and also Diane Kadonega, I have made you all co-host because I haven't seen Sarah, Eric, or Patrick yet. I may have missed their names, but so if you guys can help me as we go, kind of monitor for audio that's come off mute and make sure people are muted, just so we're not hearing background noises. And then I'll try to come back in and find Patrick, Eric, and Sarah and turn them up to co-hosts, okay? And we are recording today's session, so we'll post that as soon as the recording is available. I just wanted to share a couple quick notes before we turn it over. We're going to do updates, what we're calling Erie to Ohio updates. So what's happening at the lake and down to the river, so we get a sense of that phenology across the state. But I wanted to share, let's see if I'm sharing the right screen. Something that I'm noticing, like Ashley, just got my second COVID shot last Saturday. So we did the tour of Ohio because my husband and I both wanted to get it and needed to travel to do that to different towns, actually. But there were so many sugar maples in full bloom, really striking this year. And I put a couple images up there, that kind of chartreuse green with those long flowers that kind of dangle in the wind. So if you're noticing that, just for some reason, a really prolific bloom on sugar maples. And the day after I took the picture, I got a text from Dan Herms, and he's like, have you seen the sugar maples? They're really something this year. So, so keep your eye out for that. I also wanted to just thank everybody and recognize all the amazing naturalists that we have in the group. This is our project on iNaturalist, the OSU Phenology Fridays. And all you have to do is go in and join, and then you actually can just go into the project and look. Or if you join the project, then any observations you were already making on iNaturalist automatically get filtered into this project. So we have 77 people, I'm assuming all from this class, who have joined and are observers, and already we're up to almost 2,000 observations. So one thing that's cool about iNaturalist, so up here, I have put the link to the GDD calendar right there. So if you get in, you make an observation, try to remember to put your GDD number in the notes so that we have that kind of we can relate the date and the location also to the growing degree day units. But one thing that's neat about projects is you can sort by species. So when I click on species, I get the most commonly observed species for this project, from the most observed to the least observed. So look, we're seeing lots of cutleaf toothwort across the state, Virginia spring beauties, lots of spring beauties, bloodroot, Dutchman's breeches, and so on. And you can follow this all the way down. We have 531 species that have been observed. So it's really neat to get a glimpse of what people are noting. And I went all the way down to the bottom of the list because we have a lot of we have so much knowledge in this group, and I'm really thrilled about that. We've had folks who have identified spiders and flies and turtles and snakes and fungi and all kinds of plants. And so really want to thank all of you for putting those observations up there. Remember, you don't have to identify them. You just put an observation up on iNaturalist, and experts will help to identify it. And then you have kind of a life list on your phone of what you've seen over time. Okay. And at the end of this session, if you want to hang out around after we formally adjourn, I'll help anybody who wants a little guidance for getting started with iNaturalist. It's not difficult. I'll give you a little quick tour. You can stay for 2 minutes or 10 minutes, and that'll be at the end. Okay, so I'm a taskmaster today, and I have my bell. But I'm on a delay, right? So maybe you hear my bell before you hear me say ding. But I'm going to try to keep us on schedule because we have lots of sharing to do. And we're going to start. I asked Mary Brennan and Kathy Gunn to give us our Erie to Ohio update. Mary living up in Chardon and Kathy down in Cincinnati. And Mary, I wasn't able to change the order of your slides. I'm sorry, we didn't get to coordinate that, but so I'll turn it over to you, Mary.
Okay, good morning, everyone. and welcome to, I would love to say sunny Chardon, but I can't say that. But I just wanted to give you a quick overview of what I'm seeing up here. And one of the things that I found interesting, because we are right next to this wonderful lake, we have a lot of levels of terrain up here, and I just happen to be up in the Munson Chardon area. So I thought, in going over what I've been seeing, I'd kind of give you an overview from Chardon then down to the lake, because it certainly seems as if the different elevations have a lot to do with what we see. I've noticed that you're up here in Chardon and things are early spring. We're at the elevation of 1,290. And I would say, starting from up above, the tree canopy, thank you, Denise. The tree canopy is only about 30% filled in. And you can see with these pictures that I took out in my backyard that last Saturday, as a matter of fact, that things are just really starting. That's my beautiful serviceberry there. And I would say it's kind of at first bloom. And again, this was last Saturday on the 10th. Cutleaf toothwort, the little guy over there in the corner is the jack-in-the-pulpit, just kind of waking up. And then, of course, our May apples. The red maple has pretty much shed its flowers by now. I mentioned the serviceberry. The other thing, and I don't have pictures, well, I do have a picture right here, as a matter of fact, of my wisteria, which is really phenomenal this year. And I'd love to hear if anyone else is having any luck with their wisterias. It has not bloomed for several years. This year, it is absolutely covered with those early blooms. The middle picture there, you can see is our peach tree that's lovely in bloom. I will mention, though, none of the crab apples have bloomed as yet. And then, of course, there's the trout lily. So we do have a Japanese maple out front, and on the far left, you can see that picture was taken again last Saturday. And you can see it's just starting to open up, whereas the middle picture I took on Wednesday. So there's quite a bit of difference there. And just to mention the difference in the GDD here, on the 10th, it was 196. By Wednesday, this would be Tuesday, it was 212. By Wednesday, 214. So you can really see the response to some of those lovely warm days. Down in the lower left, you can, thank you, Denise, you can see the serviceberry, and it has come into full bloom now. And even as I look out back now, it's starting definitely past full bloom, although I'm sure the rain has had a lot to do with it looking so sad. And Jane, this is for you. On my far right, that's my pawpaw tree, and you can see it does have buds on it. I'm just going to mention very quickly, because I will be going over to tell you what I saw at Holden, but they have a pawpaw grove, so to speak, over there. It's not leafed as yet. It is absolutely, if you look up, and you can look up into the trees, they are covered with pawpaw blossoms. And I find that interesting, because I know that last year we also had a bumper crop, if I can say that, in the pawpaws. So I guess this is another mast year. So now we'll go down to Denise, I'm going to ask, would you mind going back to the very first slide with my name on it? Thank you so much. These pictures were taken on Tuesday at Holden Arboretum. Holden is pretty much in line with the lake from where I am here, but it is at 990 feet. They are much further along in things being growing and sprouting and emerging. Their GDD is not dissimilar from Chardon's. It's off maybe only by 1 degree here and there. So I was thinking, well, why is it so much further along? I'm sure the fact that it gets tender loving care has a lot to do with that. Also, the fact that the density of the growing mediums and everything, so that may have something to do with it. But those are some of the blossoms that I had seen on last Tuesday. I also noticed at Holden that their canopy, if I hadn't mentioned this, my apologies, is about 60%. Their fruit trees are blooming beautifully. They do have some deciduous conifers so that the larch is fully open. The dawn redwood is about 20% open, and that poor old bald cypress, they're just still hunkering down. They're not doing much of anything. Not necessarily in this picture, and I'll get to this picture in a moment, but as far as some of the other things that are in bloom at Holden, as you can see in the center picture there, their quince is fully in bloom, as that wonderful bumblebee is enjoying some nectar there.
Mary, my singing bowl is giving you the 30-second wrap up. I'm sorry.
30 seconds. Okay. So I'm going, you can see the pictures of Holden mentioned very quickly down in Mentor, which is at 690 feet. Their canopy is about 80%. I'm going back to Holden. I want to mention a couple of things in the picture. The upper left-hand corner is a rarity in Ohio, and that is the Lakeside Daisy, and they are all in bloom. The dogwoods are just starting to open up, and I'm not sure if this is a good thing or not, but the lilacs are blooming, and that's not necessarily a great thing, but there they are. Normally, lilacs don't open until 2:34, so they are definitely early. And I will now be quiet.
Awesome. Thank you. Thank you so much, Mary. Nice update. And I love that you went to 2 different locations to kind of compare the two and add the elevation in. Now we're going down to the river with Kathy Gunn. Kathy, I forgot to mention Mary and Kathy are both volunteer pollinator specialists and active naturalists involved in OCVN. And Kathy, I'll turn it over to you.
Okay, I've never done this before, so let me know if I can do something differently.
I'll give you a ding when we're 60 seconds, so you.
Perfect. I really enjoyed Mary's talk. In fact, her the slide with her name on it was very similar to what I saw yesterday when I was hiking in the woods at Cincinnati Nature Center. But starting with this first slide, this kind of shows what I first start to see is my first impression of spring when I walk through the woods and the trees are pretty bare. The first bee I saw this year was on March 6th, and it's the spring beauty, which is the middle slide there. That's a spring beauty mining bee, a specialist bee that hangs out on that particular flower. And I think next to it is Denise's favorite bee, I think, is Caletes. It's a little polyester bee. She lines her nest with like a plastic-like substance. And you can actually, at first I thought it was spider web in the lower left corner, but I was looking and I was like, oh my gosh, I think that's actually the plastic that she lines her nest with. I was really happy to have gotten that picture. So that was kind of, like I said, March. And then I believe the picture in the middle there of the spring beauty bee was March 23rd. And I was out yesterday and really, really surprised that the trees were leafing out and the spring beauties were fading. So the other thing that I've noticed is a lot of nesting behavior, and this is a blue heron rookery that we have at Long Branch Farm, which is about 20 miles away from Cincinnati, and it tends to be more aligned with where you guys are, so it's not as far along. We started last year with only 2 or 3 nests, and this year there are plenty more. They've also been seen along the Little Miami River, which is very close to my house, as well as a bald eagle's nest, which is along the Little Miami River. So that's really, really exciting. And finally, this is, I'm seeing, and when I was out yesterday, I saw, and unfortunately, I don't have the slides from what I saw yesterday when I was out. I am seeing the queen bumblebees are out, particularly in the forest, just doing exactly what you'd expect with, over the leafy debris. They're just kind of flying around looking for nests. It's really hard to capture them though, because they don't really land on flowers as much. I did see bumblebees really enjoying cutleaf toothwort. So, but that again is fading as well. What I am seeing now though, the red buds are really, really blooming. And I'm not sure if that is a red bud specialist bee, but I'm doing the specialist bee project, and I was really excited to sort of get a picture of one. Seeing a lot of bluebells, and I actually wrote down what I saw on my hike. The hardwood trees are leafing out. I saw a Ohio buckeye tree that looked like it was, at least probably more than 50% fully bloomed out. Seeing a lot of blue phlox. I love celandine poppy, and that's where my impression was very similar to what Mary was seeing in her first slide, which I thought was really unusual. Okay, squirrel corn, larkspur, bluebells, and Mayapples kind of have their leaves but no blooms. So I would say that's, this is a slide by a friend of mine, Jim, and I couldn't believe he captured this beautiful, beautiful nesting behavior by bluebirds. And there's actually a series that he took that I just love. So I think we're getting full on summer. I'm not expecting to see many bees in the forest anymore. I'm going to start looking more in the sunshine in the fields. That's it.
Wonderful. Thanks, Kathy. Thanks, Mary and Kathy, both of you. I know it's hard to only spend 5 minutes or so talking about all the things you're seeing when spring is so active, so many insects and flowers out, but really appreciate that kind of perspective, what's happening across the state. We'll do that in each of our lectures, get a sense of north to south. So Bob, Dr. Bob, if you want to try to share your screen, we can switch over to you. And so Bob Heath is a retired professor from Kent State University in the Department of Biology. He's a volunteer pollinator specialist as well. And when I asked him if he would do history shorts, 10 minutes on some phenology history for each of our lectures, he said, I'm a professor, I can't stick to 10 minutes. So Bob, I do have my little singing bell here. I'll give you a 60-second warning that we're getting close, but thanks so much, Bob, and I'll let you take it away. If you're speaking, Bob, I don't hear you. Are you muted?
Can you hear me now?
I can hear you now. Thank
Great.
you.
Now, one of the longest phenology records is the records kept in Japan of the time when the cherries come into bloom. And it's important in Japanese culture to see this. It's a big event, and it's what's nice about it is that it's a very short-term event. And so timing that, saying this is the day when that happened, is really kind of important. And what I'm going to talk about here is, instead of talking about the cherry blossoms, I'm going to talk about the value of having long-term records. And so we'll talk about the past, present, and future. There we go. The species of cherry is Prunus Yedoensis, and the variety is the Yoshino cherry. And so they have kept records of this for a long time. And recently, someone took all those records and plotted them out. One of the values of having a long-term set is to say, are there any trends in this? You can see these are the early blooms down in here. These would be the later blooms. And this is starting at 812 AD, and it's then moving. The right in here would be the what's called the Little Ice Age, where the Earth, there was a chill over the whole Earth, and especially in the Northern Hemisphere. So you can see that it's really pretty even until pretty recently. And then it's starting to trend down. This is this year's datum. And so one of the values of having a long-term data set is that you can look for changes in the long-term trends. And so this would be a very good piece of evidence for global climate change, that it's warming up and therefore the cherries are blooming sooner. Or to put it into our terms, the growing degree day would be coming earlier and earlier in the calendar. Well, the cherry, the Prunus yedoensis Yoshino, is also the same cherry that the Japanese gave to the United States as a gift, and which is planted in Washington, D.C. Now, this is a picture I took when I took my grandkids there in a couple of years ago on April 1st. So you can see it was in full bloom and it was beautiful. And even my teenage grandkids appreciated it and loved it. One of the things that you can do with these long data sets is you can also model it. And this is out of a paper that you can get for free because it's in the Public Library of Science 1. And what they did here was they said, well, since we have a long-term data set, we can make a model of it. I'm not going to go into that except to say they tested the model, and if the prediction from the model equaled was exactly correct, then it would lie right on this dashed line. And you can see that many of the points lie very close. And for those of you who are statistically minded, the R squared on this linear regression is 0.62. So that's actually pretty good. Well, now that you have a model, how can you use that? You can use a model to forecast. And so what these investigators, Chung et al., did in 2011 is to say, well, this is the average date of the peak cherry blossom dates in Washington, D.C. You can see the legend is over here, and you can see the pinker and redder it is, the later it is. And it goes yellow, green, and then blue. So you can see, well, okay, this is what it was as an average over the past 50 years or so. But using global climate change models, they can also forecast what will happen to the peak cherry blooms. And this is what they can predict from those models. You can see that the models are different. This is the IPCC model, and this is the MPI model. And they differ, but it's interesting to see how you can use the cherry blossoms as a way of looking at, or at least being able to forecast how the terrain and the blooming of the cherries would progress. And this is, let's see, this goes up through 20, 2070 to 2099. Excuse me. So that's it. There are the references, and Denise is recording this, so you can check out the references if you wish. I think the BBC News article was one that everybody read in preparation for today's thing, and then this is the article in +1. So that's it. I think I did it, Denise. I didn't hear you ring the bell.
Bob, you get 2 rings for being so timely. Thank you.
Okay.
So next week, Dr. Bob is going to take us into the world of birds. Easy for me to say for our history short. So look for that on our week 3 materials. And great. So Ashley, if you want to go ahead and share and see if that's working good. It's I'm really excited to have Ashley Kulhanek with us this morning to give our lecture talking about her work as a master's student in the Department of Entomology, which is where I first met Ashley. She was working with Dr. Dan Herms in his lab, and I think she's going to tell us about her graduate work and then how she's transitioned into the Ag and Natural Resources educator position in Medina County and how phenology shapes what she does there. Ashley, thanks so much for joining us. I don't know if I have to give you the gentle reminder, but
you probably will.
Excited to have you here. Thanks, Ashley.
So, hello. Hi, guys. A lot of you I've probably met in the past because of my work with phenology, but I will share a little bit about my past and my history because it really was an accident. So an accidental passion that I still use today. So, I was a sociology major at Balmoral College. I took an entomology class at Stone Lab. Here I am with my little bandana. Back in undergrad, and I fell in love with bugs, and I'm like, I'm going to be an entomologist and I'm going to study bugs, and this is going to be my new thing. And so I switched my major and everything. And so then naive little me, loving bugs, is like, well, what do I do? Grad school, let's do it. And so I joined the Herms lab. Here's Dan Herms up here. You may have had him as a speaker before or already, or he's coming up. So I know you'll have him for this talk. And it was like, all right, I'm ready. What do I do? And, those of you who've been to grad school, you think, oh, I'm going to catch all these bugs and it's going to be great. But it's, you have to have a project and thesis and a research study. And so I went in thinking I'm going to save the ash trees. I'm going to develop an ash tree that's going to resist the emerald ash borer and save the day. But I was an entomology student, not a horticulture student, and I did not realize, it takes decades to develop ash trees or any, horticultural variety or cultivar that's going to have different traits. And so, that wasn't going to work. And then we said, maybe we could have dogs sniff out the larva in logs and in trees and products, and then we can prevent it from spreading. And, that idea was brought to us. And I'm like, well, I'm a cat person and I'm not going to raise dogs at my house and train them because, where were they going to live? Who was going to take care of them? And so that didn't work out. And we talked about, okay, maybe some dendrochronology. But all this time, I was working on the phenology project. One of my research assistant duties for Dan was to update the phenology calendar website. And I worked on putting fact sheets and pictures into it with different information. And this was back when the website looked like this. Look at that great old branding. And so I put fact sheets and photos and, we were keeping it up to date. And I'm like, Dan, I've been working on this project for you in the summer. And, it's really interesting me now that I'm kind of understanding it and learning about it, but nobody was doing anything with the data. Denise and Dan had started the Phenology Garden Network in 2004 with gardens across the state, monitoring plant blooming, first in full bloom, which you've learned about, and how it relates to horticulture and pests and insects. And it was a way that we could start collecting some of that data for potential climate monitoring, as Bob mentioned, and to use it for extension education, which I was learning more about too as a grad student. And so those 28 sites became 36 gardens, and you as master gardeners, this was my first introduction to a lot of master gardeners and certified volunteer naturalists, we're collecting this data on the 16 clonal cultivars of woody ornamental. And as you know, the gardens grew to include perennials and then native plants for pollinators, and then we were recording pollinator data. But back when it started, we were just focusing on these woody ornamentals. And so the two major questions I was tasked with answering was, is the growing degree day model that we were using, which was a generalized model using a start date of January 1st and a base temperature of 50 degrees Fahrenheit across the board for every plant, for every pest, was it good enough to use to kind of place the sequence in that calendar? And it was. And then our biological calendars consistent year to year and location to location in Ohio. And we were hypothesizing that the phenological sequence that Dan had developed at Seacrest and 5 years at Dow Gardens in Michigan was going to be consistent across the state so that people in Cincinnati down on the river and people in Chardon up by the lake could still use that sequence of blooming and insect emergence to make pest management decisions and help predict when things are going to happen. Does it change by location or, how far south or north we go? And so we did some tests to see if the sequence was going to remain the same garden to garden. So here's a look at Worcester versus Summit County's garden in 2008. You can see that the sequence lined up almost perfectly. We had instances where there were ties where two plants would bloom kind of at the same time or were recorded at the same time by the volunteers. And there's a lot of variation in that, like were you going every day? Were you going once a week? How were the volunteers visiting those gardens location to location? So there was variation there. And sometimes there were flip-flops where, one gardener might report that, the elderberry bloomed, 15th and the Bumald spirea bloomed 16th, but then reversed that in a different garden. So these gardens are really close together, Summit and Worcester, but when we compared further and further away, we still saw that there was a lot of similarity between the sequence of first and full bloom in these plants. And so we found that based on the data we had at the time, that these are pretty reliable year to year and location to location. So again, here's some of those ties and flip-flops, which could be related to microclimate. It could be related to elevation. Those are some of the things we didn't assess for. Mary mentioned that, there were some elevation changes around Chardon with her reports. And so those things could have factored into the sequence of these blooming events in these gardens, but it was something we didn't look into further because I graduated. But then there was one case where Richland County's garden just never worked well. And part of it was a lot of the plants died or the deer got to them. And so we didn't have a lot of data points to compare, but there were some instances where we're like, whoa, this doesn't work. And so we could chalk it up to, the plants weren't there. Mansfield's kind of in a little bubble. And so it might be an elevation issue, but all in all, we were able to find that for the most part, we saw really consistent sequences from year to year and location to location. So yay, go ahead and use the growing degree day calendar. And so I was doing these updates for master gardeners and volunteer naturalists who were collecting this data throughout my thesis and defended. But then, so like many graduate students, what does one do with a degree in an obscure science within an obscure science? So I had studied phenology in entomology with bugs and plants. And I'm like, well, what do I do now? But of course, as a grad student, I did a lot of outreach and extension work. So working with Denise, working with you all, teaching and programming about what I was studying, as well as, working with Dan's lab and Emerald Ash Borer. And so it was such a great natural fit to kind of go and say, okay, let's talk about extension and working there. So the first step is, of course, to make your obscure science totally awesome and get people excited about it and teach it so that it's not obscure anymore. And so this is the Discovery Pavilion at Seacrest Arboretum that has a lot of information about phenology in it. This is Denise speaking to Master Gardeners here, this picture. So, being able to spread the word about phenology was one of my first goals, because you come out of grad school and you're doing teaching, well, you teach what you know. And it came full circle. I became an extension educator in agriculture for Medina County, which is my home county. And I was shocked and appalled to learn that all this data I had been analyzing, there was not a phenology garden in Medina during the years I studied. And there was no plans to put one in. And so here I came and I'm like, we're putting a phenology garden in because, this was my passion project. And so our master gardeners in Medina installed it, and we put in a phenology garden, which has grown and now includes pollinator plants, and it's become more of a pollinator focused space, as well as including some of those original plants. And it's been a great opportunity to teach from, both for our master gardeners when training new master gardeners, and also for educating the public about phenology, monitoring plants, as well as the pollinators that visit them and why they might want to plant those plants there. But as an extension educator working with the public, what am I doing with phenology now? So one of the things we use it for is to actually help people plan gardens. This is one of the activities we do when we train master gardeners for garden planning. But we also talk to the public about how they can use the sequence of blooms and knowing when they bloom throughout the season to plan a season-long blooming garden, or if it's for bees, if we want to feed the bees all season long, we can use phenology and those blooming sequences to kind of see what plants are going to bloom when and for how long and in what sequence, so that we have something interesting happening in the garden all year long. So that's one of the more fun and kind of fancy free ways we can use phenology. But then also for pest management, which was what my research was really focused on, was using those biological indicators in the phenological calendar as indicators for pests, because pests, whether they're insects or weeds, can be hard to time. They can be less visible, harder to notice if you're someone in the general public who hasn't been trained to look for them or don't have the time to be out scouting day to day. And even a lot of landscape companies and exterminating companies, they've trained their people on how to do the tasks, but they might not have the background to identify or observe insects and know what they're looking for. So one of the ones I've been talking about just recently is using Forsythia to time preventative crabgrass control. So Dr. Cardina in the Horticulture and Crop Science Department at OSU did some work using growing degree days to predict when the germination of crabgrass seeds was going to happen. And it found that just as the Forsythia is starting to kind of give way and look kind of cruddy, that's a good indicator to make sure you've put that preventative treatment down to make sure you catch those seeds before they emerge. And so smooth crabgrass emerges before large crabgrass, and it emerges slowly. Sorry, my little clicker's not working. Turn off the laser pointer here. But I also wanted to point out that if you miss the window for your preventive treatment, Dr. Dave Gardner in our turf program says that this new Roundup crabgrass destroyer works pretty well to get it if you miss the preventive treatment. So just a little tip for y'all. But smooth crabgrass emerges slowly when the soil temperatures are getting up above 54 degrees. And then we can use the bright yellow flowers of the Forsythia to kind of figure out when that is going to happen. Now, the trouble with Forsythia, as many of you know, is that sometimes the blooms get frozen off. And this has been one of the first years where I've seen it beautiful, full bloom Forsythia. And for a really long time, they haven't really gotten that damage and started to look cruddy. And so that may or may not give a false sense of, did you get that crabgrass prevention down in time, because you've had this huge window of blooming for Forsythia. So we'll see how that goes this year. But also for some serious insect pests, especially the ones that are really hard to see, like scale insects. So this is calico scale, and it's a non-native invasive scale. And the adults are big and puffy. It's a soft scale, so they produce a lot of honeydew. They can have their population sneak up on you because they lay a lot of eggs. And it feeds on the phloem of the plant, lots of fluids going through them, so they're big honeydew producers. Lots of hosts, buckeye, crabapple, dogwood, honey locust, magnolia, and on and on. So they can be a pest on a lot of different plants in the landscape as well. Like most scales, they don't kill a plant outright, but they weaken the plant. That makes it susceptible to other environmental and insect and disease pressures. And so damage can sneak up on them really fast. So it's weakening the plants, and then over time we can see them decline and potentially die from a combination of factors. And so this is from Joe Boggs, who's down in Cincinnati area. He took this picture of a honey locust that was in an island in a parking lot, and so those are terrible places to plant anything to begin with compaction, poor soil, not a lot of root space. But it was infested with calico scale, and so in 2014, this is what it looks like. And then in 2015, it was dead. So this was a combination of bad planting location, heat island. You're getting a lot of that radiant heat off of the parking lot. There's not good soil there probably. Plus, it had calico scale and potentially other pests affecting it. And so this is a really bad outcome. So how do we manage calico scale? Well, Dan Herms did a study that found that the best and most efficient management of it is going to be when we target the summer crawlers. And anyone who's not had the fortune of capturing these tiny, tiny crawler stages of a scale. So you see these big, puffy, waxy covered bodies of the adult scales, but when the eggs hatch and you have these little crawler stages, they're teeny, weeny, teeny, tiny. And you can see here in this picture, along the leaflet of the honey locust, you can see these tiny baby yellow scales. And so this is a leaflet of the honey locust. They're so small. And unless you need to be out there looking for it and have seen the calico scale in the first place to look for the crawlers, it can be a really hard stage to catch before they start excreting that waxy coating and becoming more difficult to control. So we use phenology here. So the eggs hatch around 740 growing degree days. And so if you like using the growing degree day calendar, you can check back and keep track of where your growing degree days are in your area. Or we can use a phenological indicator, a plant bloom, to represent that growing degree day temperature. And so when we talk about calico scale, we might look for northern Catalpa in first bloom, Weigella in full bloom, and Forsythia. So these are coming out right before egg hatch. And so when you start to see these in the landscape, you can then know to start scouting. So it's not saying, okay, as soon as northern Catalpa comes out, you want to start spraying, but you can start scouting for those vulnerable stages of the calico scale. And so then we can talk to landscapers and homeowners about, if you've had trouble with certain pests, what could you plant in your landscape and why would it be beneficial? So northern Catalpa, I think it's beautiful. It's native. One of the benefits of this also is that I don't necessarily need to plant one in my landscape because at least here in Medina County, I have plenty of examples. It's a very easily identified tree as it's the only native with the whorled arrangement, those big beans, the big, beautiful orchid-like flowers. You don't have to plant one, just drive around until you see one in bloom. And then you can know, all right, it's timing for calico scale. I've had a history of it or problems with it in the past. That can be a reminder to me to start scouting. Forsythia, I have never heard of this in my life before I started looking to different indicators. And so it is a non-native, but it's hardy in our area. It has a big growth pattern. So if you want something that can be like a little bit of a hedgerow, you could use that. Weigella was one of our original 16. The Red Prince variety was one of the original 16 in our garden. on our phenology garden. So this could be a great option, great for hummingbirds. And so having those conversations with homeowners or landscapers about, well, why pick this? Well, if the homeowner's looking for something red, they're looking for something for the birds. And it can also help you with any pest management, or them with any pest management they might have to deal with. And so, we have those conversations about how these plants can help and how they can use the calendar if they just want to go and look up the growing degree days day to day without using those biological indicators. And it's a great way to have a conversation about improving those pest management strategies and saving time to not have to be out there scouting every single day if you're experiencing pest management problems in your landscape, especially very diverse landscapes. And so another example would be spruce spider mite. We have a lot of trouble with homeowners and landscapers alike. You see the damage of spider mites kind of after they've already been there. You start seeing that bronzing, that stippling damage on the leaves and the needles, but they've fed already and oftentimes they're long gone. If we go in and spray now, usually we're damaging beneficial predators that have already come in and started feeding on these mites. And we end up with a rebound population by killing off all the beneficials, trying to control the pest, which has already done its damage and moved on. So, it's a cool season mite. It causes this bronzing and bleaching, especially in some of our evergreens, which then those needles are going to stay there and it's going to look sad for a while. It can cause this browning on all of our spruce and arborvitae and junipers. And so we can look to serviceberry and Callery pear to be indicators to look for those mites now that they're hatching. Again, egg hatch is not necessarily the best time to spray. You want to have that window where they've all hatched and started feeding so that you can apply any pest management strategies you need to. And then you can go out with a white sheet of paper or a cake pan with white paper or towel underneath it and bang the branches. And then if anything falls onto that white paper and starts scurrying around, you can smear it, you crush it and smear it. And if it's got green to brown coloration, it's likely a pest mite. And if it's got a more red coloration, it's a beneficial mite. And so that's actually a scouting technique we use for mites. So then you can go out and check those trees if you've been experiencing problems or you're concerned about it, especially in years where there's high stress or if it was just planted, where it would be vulnerable to that kind of damage. And so this has been one of my favorite things to talk about with people is how we can use biological indicators and the phenology calendar to better predict those pests, because people who call and say, how do I control it? And if you just give them, use this product or that product or increase these beneficial insects, if it doesn't work because their timing is off, then they think we can't do our job. We've given them bad information, and it may just be that they've timed it very poorly, especially with crabgrass. It's like everybody misses crabgrass. It always gets away. And so this has been a really great tool for me to work with the community about pest management. And because, again, I have my passion for entomology and insects too, the hope is we're doing the least amount of damage to the beneficials by timing it very specifically to the pests that we're having issues with, and hopefully doing less damage to the beneficials we want. Again, other examples, bagworm, two-spotted spider mite. Bagworm's really obvious when it's a big fourth instar caterpillar, and these big cocoons that they've built are very visible and look like pine cones. But the best time to control them is when they're very, very tiny and they only have a little dunce cap on their head. That's about how big their cocoon is at the time that's best used to treat them, especially with BT or Bacillus thuringiensis, which is natural occurring bacteria that can kill caterpillars of the Lepidopteran, and then it doesn't have as much of an impact on our other insects. And so again, Japanese tree lilac, that is becoming a really popular street tree. And so again, if you don't have one in your landscape, you could probably drive around and find one and use that as an indicator for egg hatch of two-spotted spider mite and bagworm. And then you go out and start scouting for those pests if you have issues with them. And then finally, I'll bring out the new invasive species. So we've had some great luck using biological indicators and phenology for invasive species. So we use black cherry for gypsy moth spray. We use the black locust for emerald ash borer. So what about spotted lanternfly? It's new. We just found it in Ohio. And it's fairly untested with the phenology and indicators out there. There was a study that was just published last year in April 2020 about their growing degree days in their development. And so they found that egg hatch and that first instar is found on average around 270 growing degree days. So the range was 228 to 321 growing degree days in their study, which took place in Pennsylvania. And they used a 50-degree base temperature, which is what we use for our calendar. What wasn't clear is whether they started on January 1st for their calculations. So I'm just assuming that's what they did. But if we're looking to scout for that egg hatch, if we're going to catch them before they disperse, before they grow and become adults and have the ability to glide with their wings, we can start looking for them around 270 growing degree days. Now, because this is just one study, I think they had about 4 or 5 years of data. We don't know if that's necessarily going to be the magic number. And so this is something we can try and test, especially as we're out there trying to monitor it in our areas. If any of you are from the Jefferson-Harrison area, I think it was Jefferson County that they found it in. And then also if you have areas where there are grape wineries, which is where the big concern is in the industry. And so we can start here and then tweak it to see how it works for us. So if we take a look, you know that early in the season, a lot of things are happening and they can happen very quickly. So if we're looking around 270, but with a range of 228 to 321, we can look at flowering dogwood, first bloom of red buckeye and horse chestnut, rhodosier dogwood. And so those could be indicators that could help us with timing for spotted lanternfly. Or again, if you are often using the growing degree day calendar and are looking up your growing degree days day by day, as many of you are in your journaling, you could just use the actual growing degree day count and not rely on the indicators. But this is something I'm excited to see how it works and see if people find an indicator that works best for them. And the study also talked about the second and third and fourth instar as well as adults. And so we can find those growing degree days in our calendar already, even though it's not posted up there, but eventually as it's been tested, we could add it to our calendar here at OSU, and just like emerald ash borer and gypsy moth have been. And then lastly, it's been fun to talk about phenology with farmers of traditional agricultural crops. So everything we've talked about has really been landscape focused. We're talking about ornamentals, we're talking about ornamental pests, and that's what the original calendar was designed around. We used data from the Arboretums at Dow and Seacrest. These were varieties and cultivars that we were studying the stages of, and all the pests in the calendar are ornamental pests. And so how do we maybe use this for people who are growing vegetables or growing soybeans? And so we don't have a calendar for that exactly, but a lot of what's already a common practice in agriculture is phenology. So for example, using the stages of soybean to time scouting for pests. So this is late V stage for soybean and R1, which is reproductive stage 1, when those flowers are just blooming. And that's the best time to scout for stink bugs. And so they're using indicators, phenological indicators in the actual crop they're growing to decide when to go out and do sweep net tests for stink bugs, and then using a threshold. If they find 4 bugs on average per sweep test, it's time to spray for stink bugs in soybean. And so that's one that's been really tried and tested and true. But we also talk about with farmers and vegetable growers about how they can create their own calendar. And most of you are doing that by journaling the activities in your area. So, you're out there scouting for your pest, and you can note what's in bloom out there. Do you have a tree on your property or a shrub that's blooming? What stage is your crop in at the time you find the pest? And see if that can help you to be an indicator as you manage the pest into the future. And then try it, test it, and see if it works. And if it's not that reliable, you go back to square one and see what other plant or growing degree day works best for you. And so we are encouraging farmers that might be having trouble to try this out in their own farms and see if they can use phenology to help them. And just for fun, I found that Facebook and Instagram have also been kind of a great phenological calendar, because people are posting pictures of when different plants are emerging, and especially ones that don't exist in our calendar. So this is skunk cabbage, and it certainly isn't on our phenology calendar. It's not one we've studied before, but here I can kind of keep track of, they saw this on February 11th. And then what was happening at that time. I can go back into our growing degree day calendar, change the date to February 11th of, this was 2020, and say, what was the growing degree days at that time? And is that reliable for predicting skunk cabbage each year? And so using the observations of each other and then sharing them, whether it's on iNaturalist or Facebook or Instagram, you can kind of find where things and when things are blooming that might not have already been studied or recorded. Also, you all are a little bit of a phenological indicator. When we start seeing spring workshops from master gardeners at OCBN, it's kind of the sign of spring. And so all of a sudden, all the programs start coming out. And also our Buckeye Yard and Garden can be a little bit of a phenological indicator, because most of our writers are out there observing just like you are. And when an event happens, we write an article about it and often include the growing degree days and what else is going on to help manage it. So just like we talked about the calico scale, oftentimes Joe, who is down south, is the first to catch those crawlers in action, and he'll put an article up, and that can be an alert to everybody to start scouting for it because it's on its way. So great use of phenology in my job, and really brings me a lot of joy because it's something different and it can be really uncomplicated. We don't have to do the math ourselves. We can use the plants, we can use the calendar, and we don't have to be too overly burdened by scouting and timing for our pest management if we use the tools at our disposal. So that's it for me, guys. Thank you. And let me know if you have any questions.
Awesome. Thank you so much, Ashley. I love how you bring it all together and show how you've used phenology as a tool in your work that has so many different applications in a lot of different audiences. I remember when you were a brand new educator and, kind of struggling with, I know a lot about entomology, but how do I help farmers? How do I help, 4-H families and master gardeners and so on? And I just think phenology is this great tool. Many of our participants are naturalists and as well as gardeners and or in other professions, and it's just such a portable tool that can help you in a lot of different areas in natural history. So
absolutely,
yeah, I appreciate
Thanks,
that.
guys.
Ashley, there was a question, and if you have any questions, folks, go ahead and put those in the chat box. We have a couple minutes for questions. And I've wondered about this too, and maybe someone in the audience, if they have this resource, could put it also in the chat box. But Ashley, do you know of an Excel application or some way to kind of expand? I've always wanted this tool, like how can we take our calendar and put it in a way that people can add their own observations in and still have kind of that sequence or that scroll? Do you have a tool or an idea, a thought on that?
So I too have been, struggling with this because I want to be able to edit the calendar, but then it risks the integrity of the calendar that has been actually recorded years and years and years from Dan's work. And so there's not a tool that we have yet, but this is something we can work on, Denise. I mean, we can copy it and put it into Excel and then make it available to us. There might even be a way to make kind of a second calendar that's not quite called that, so it doesn't come up in the search the same way, but kind of like you've created the iNaturalist phenology study group, if we could have one that we could play with, because I often teach this topic, and then of course the people who are growing vegetables and growing crops, they want, well, where's my calendar? And it just hadn't been made yet. It hasn't been studied yet to put one officially together. And so we talk about people making their own, but why can't we make our own together where we can all contribute? And that'd be a great tool that we could work on.
Yeah,
I'm in. Let's do it.
We have actually on our so Dan is coming on our very last time together on May 7th, and Dave Lonis is going to join us as well. So Dave Lonis is the genius behind the calendar, the mechanics, the technology of the calendar, incorporating the weather station data into what we see online. And this would be a great question for him, because he probably, we give him the problem, he can probably help us think through how to create a tool, an app or something. So
yeah, and Tissa, she put that, there could be a mobile app for it, which would be fun. So,
and I'll share, I have an Excel file where I've taken the calendar and put it into an Excel file. And then I add my observations in, but the challenge is when you're seeing a lot of things at the same time, do these go beside each other? Because you can't have this infinite scroll, because it's hard to tease out what's next and what's next and what's next. But it's more like there's this block and then this block and then this block. But yeah,
right. And that's what we saw when we did the study, is that there was a lot that tied, or that they were just flipped. And there are other factors that we don't account for when we do phenology growing degree days, like elevation and stuff, and they're just not there. And some plants just don't work well with it. Some are more related to photoperiod. So later in the season, you kind of lose some of the punch of it. And so also we found that full bloom is kind of the hard one, where we just don't have a good grasp of nailing peak or full bloom. And so the first bloom is what's really more beneficial when we're looking at phenological indicators. And so maybe we can make one that's just full bloom, or first bloom rather, and make it simpler instead of having, all of the indicators that are in the current edition.
Right. Good point. There are a couple questions about phenology gardens. How do you get one started, and how do you put one in the ground?
Well, I mean, now it's kind of rough, because I don't even know, Denise, are we still collecting data the same way? So you're not really putting a phenology garden in. You would be just putting a garden in with the same indicators we've already used. And so if you certainly want to, just find a partner for the land, or if your extension office has land, to install one, or work with your parks. And then I have a list of the original plants that were there, as well as what was added. So we have a little brochure of all the species that were put in and when they were added. And you can pick and choose from those, or you can just make your own. It doesn't have to be what we did, because we're no longer collecting that data for research purposes. And it kind of morphed into kind of a phenology and pollinator garden, where we were looking more at when are these pollinators and bees emerging, and what are they landing on. And so its purpose changed, but you can still plant one and monitor it for yourselves and for your counties and your groups, and contribute to iNaturalist or anything else. So
yeah, when Dan Herms moved over to Davy Tree, folks, we stopped gathering that data. I didn't want data to come in and sit and no one was using it. So many of the gardens continued to enter their data through the National Phenology Network, the NPN. Others transformed, as Ashley was saying, into more of an educational demonstration garden, where they still teach about phenology or about pollinators, but we don't have an overall program of phenology gardens now. But that, I love, the idea of being able to teach, show those indicator plants. It's funny, Ashley, I'm sure you get this too, but people think, why would I want a phenology garden? I don't want all those pests in a display garden. But it's not like we're showing, we don't have the plants that the pests eat. We have the indicator plants, right, that remind us, when you see this blooming, be thinking about this pest. So it's an opportunity to learn. So great, Ashley. Thank you so much. I really appreciate
You're
you
welcome.
being here and sharing your expertise. Folks, we're going to shift gears to our a little section we're calling Phenology in Practice. So I asked Jean Poremski, longtime master gardener, horticulturist, and naturalist, to and nature journaler, to share how she's used phenology in her life and her profession. And Jean, do you need to share the screen? And I'll bet, are you muted, Jean?
I got it.
There you go.
Okay, not yet, Denise, because I just want to talk about all of the parallels that everyone that is on this Zoom call probably shares with you and me, you know, what we've seen, judgments we've made, because we grew up in the same part of Western Pennsylvania, just a county away, except different times, different decades. And I grew up with cement and bricks, and you grew up with nothing but forest. And that dirt road is amazing that it was so wild. Do you know what birds I had growing up? 2, Sputzies and starlings. I never heard a blue jay until I vacationed in Cuyahoga Falls. Never in all of my youth. We moved when I was 15 to 5 acres, 25 miles away, and wow. But the thing I want to say is that I started watching weather starting at the age of 9.
Oh, Jean, is your microphone maybe?
What do you think?
With rubbing, or someone's sound was off? Maybe go ahead.
Oh, wait, I have to get back on. I'm off. Oh, come on. Don't do this to me.
I can hear you.
Okay. So the fact is, I was watching weather. About the age of 9, I noticed skim ice in a tarpaulin puddle when I was going out the back door to go to school one morning. I was 9, and there was skim ice, and wow, look at that. You know, and I'd poke in it and play in it. And I was supposed to be going up the street, only 2 blocks to school. I lived in the city, the real city, Mount Washington, all pavement. And then the next year, believe it or not, on that same date, there was, well, I think the next year it was a little thicker than skim ice, November 2nd. That was so impressive on me that it started my life of observation. Sure, I played with weeds and seeds before then, but this was serious now. I started noticing weather patterns, a whole rhythm. I mean, it happened year after year after year. There could be like the first snowflakes, not snowfall. And I knew when that would happen, no later than November 4th. So these were things that were always in my mind. The big events, snowstorms that would resemble each other year after year. They'd come from the same direction. The next snowstorm would occur in the mountains, not up in the snow belt. There was a storm on around December 6th, December 8th, would be a daytime snowstorm. 6 inches of the most treacherous snow that by evening rush hour in the hills of Pittsburgh, it was chaos. I was stuck in it once with my mother driving. I didn't have a license at the time, but that stuck with me. Everything impressed me. And I kept pursuing these observations because they just stuck. They were so phenomenal. January thaws always occurred about the same week, give or take a couple of days. And I never knew there was such a thing until I heard the words and paid attention. And doggone it, if the soil didn't start to thaw during those few days in January. 2 weeks later, the coldest days of the year occurred. And that really was year after year after year. It would go below 0 for 3 or 4 days. The highs during the day could only be in the single digits or the teens. And that wouldn't moderate much throughout the rest of January, except for that January thaw. No, February was a tough one. But we had that foggy Februaries that were so depressing. And March was even denser when it came to fog. You know, the sublimation was just an overall grayness, but then real heavy fog in March. So what happened next? I didn't observe much in the spring because I was just outside playing. I mean, playing hard, all the hours outside. I was really a cowgirl. I had a stirrups and bridle. I grew out of that. I played with the old abandoned garden flowers, and in a pile of dried, curled, splintery boards that I knew to stay away from. I watched the wasp go in and out. I watched a bumblebee go in and out without knowing what was truly going on in there. But I stood and watched. I was rapt. I watched those stupid birds. My parents hated those birds. It's transferred. I still hate those birds. I have them here too. We brought all the city birds with us. I want to see if no, I didn't miss anything. Those were the rhythms. And it repeated year after year until about the 70s. And then it started to disintegrate. And I wasn't seeing the familiar anymore. I wonder, does anybody else, have they observed such in their lifetimes when they are impressionable? And has it stuck? But I moved to Ohio about the same time Dr. Herms did. So I was more advanced in age than he. Still am, as a matter of fact. And I learned through observation of his calendar and the events, they were accurate. Doggone, after a few years of our phenology garden and watching and matching up degree days to occurrences, it was true. There were no doubts. And, of course, at first, everybody's a little skeptical. It's like, show me. I said a little skeptical. I trusted him, but it was a show me. And he did. They did. Everything did. And I had client work to do. And knowing when this white pine weevil turned those leaders of the pines and spruces into those shepherd's hooks, knowing why, and there was something that could be done. And so many trees.
I'm sorry to interrupt. I think your journal, it might be on the microphone, because when you move your paper, it cut out just a little bit.
Okay. All right. I'm sorry, Denise, and I understand.
We're good.
This isn't my journal. This is just notes. It went from, what would have been about 10 pages. Honestly, this is hard work. But the white pine weevil started it all, proving the list. The calendar was accurate. Reported to my clients. They got their chemical guys on it, spray guys, not me. And they disappeared like a miracle. And they only lost one full tree because that was the one that had gotten very far ahead. After that, the Europeans pine softfly. Yeah, I learned that. I was on the lookout. The pine scale was everywhere. It was right under our noses on all those mugo pines. That was so easy to take care of. All it took was some light horticultural oil in the shade or some safer soap. I had crabgrass sprouting, and I knew people were missing the dates for putting down their pre-emergence, and they're expensive bags of multiple step product. Even now, my crabgrass has been up for my first. It's been up for quite a while. It preceded the grass seed I put down. Doggone it. And there was nothing I could do. So I'm going to have to invest in some crabgrass control or not. Then I moved on to the very late magnolia scale. And I know I related this to you because it was all new to me. I never had a magnolia. In fact, I never had any exotic trees at all. But this was a mortgage tree that came with this house. It was easy to get rid of them. I didn't do a thing. There were so many little fuzzy coverings over these twice-stabbed lady beetles, the little black ones. I saw them all over the tree, and I knew. I gave it about 2 weeks and watched. I didn't have a problem at all the next year. Nothing. And those lady beetles only increased at that time. Had I intervened, as we would have been advised, I would have ruined our beneficials. And what would it have taken for their recovery? How many years compared to the recovery of the magnolia scale? So this has all been a learning experience. I go on, when I do my radio spots, I get to communicate all of this, and I know that it's accurate. As a master gardener working on the garden information line, I could talk at length about it because I know. And thank you, Denise. And thank you, Ashley, because I've been in your audiences often. I've come up through the ranks. Now mine, let's see, now I can share a screen. Okay, Denise? Let's see if I can pull it up.
Okay. I'm going to give you like a 2-minute warning once you get it up, Jean. So we'll get your screen shared.
Oh, come on. Okay, let me get back to the beginning. Do I have another? Yeah, there's my other one. I always get stymied by the second share command. Okay, and let's go back to the beginning. All right, that's not the beginning. All right, this is messy. I have been observing so much. Look at the difference between last year's record keeping and this. COVID isolation has been very good for me because I have been out there all of the time. Last year was supreme. It taught me that I was nature. There was no division between me and everything that was out there. So I really came to terms with what ecology is. Even though I preached it, now I knew it. I was part of it. So let's look at the page close up, and I'll highlight some of the things that have been important this year. May 3rd of last year, my catbird arrived. April 6th of this year, my catbird arrived. And how I knew it was the one that was going to stay? When the 3 came the month before, I knew they were moving through. This one sang every day. I mean, it sang to me, and I knew it. It was my catbird. And on April 14th, in the back pasture line, that was only 1 week after it arrived, I found a multiflora rose bush filled with feathers. The top one was a little orange fuzzy one. The ones on the ground identified my catbird. My catbird. That was grief. I can cry right now. He'd only been here for a week. So no breeding this year. I think last year was the first year we fledged catbirds. But what can I do? 4-4, the gray tree frog appeared. I've been listening to the chorus frogs, the gray tree frogs, and noticed that I haven't had peepers. What's up with that? I only heard one peeper a few days ago. On the 8th of April, I watched the chickadees cleaning the poop out of their nest. So I knew, well, I actually saw one carrying a caterpillar in. I don't know where he got it with no leaves on the trees, but I knew they were in a stump that is only 3 feet tall. It's not even high. They're not the least bit safe. And it's only 8 feet from my compost pile. I looked inside my rain barrel to check on levels, and there was my gray tree frog, as always. I always have gray tree frogs just under the lip. This one was the biggest I've ever seen. I have starlings building their nests now with the chickadee poop. The robins and blue jays, I noted when they got frisky. I know when the mourning doves got frisky. They're so silly. They don't even look serious about sex. And the Carolina wren has a nest, I think, concurrently with the chickadee. So most of my observations, and always have been, are with birds. An Acadian flycatcher. I'm short on mason bees. All of the locations where there's only about one. But let's move on to what I do.
Another minute or so, Jean.
Yeah. Here's a composite of a lot of subjects. This is done at Phipps Perennial Garden. But then I selected some of the thumbnails, and I realized that I'm obsessed with the sex organs of plants, of flowers. And this was in California. It was just what caught my eye. Look at those bristles on the rose and the teeny tiny leaves. And the poison oak was attractive because the dead leaf was pink. But it was something that I stumbled on, appealed to me. That's why I captured it. That's how I journal. And look, this is a single subject. This is a Acer Griseum. And I really enjoyed doing this. And I found that it wasn't as much of a challenge that I thought it would be. But look, something I can share. We can plant the flower series on a page to add something to it, a record of sorts. Look, the K is the calyx. Begins with a C, but K is good. C is the corolla. So calyx is the bottom part, the sepals. The corolla is the top part, the petals. Well, I do this. And the andrecium, well, that's Andrew, that's the male. And the gynecium, no problem for us females because we have a relationship with a gynecologist. So the male and female parts. So we're counting the stamens. Look at the lily, 6 stamens. And look at the think of the lily capsule, and it's three, I can't think of the word, not locules, but carpels. So that's something you can add to your pages just so it's formulaic. And I am really worried about my hummingbirds. Last year, when they arrived, there were no red buckeyes flowering, and there was no Lonicera sempervirens, which are usually waiting for them. I don't put out sugar water of any kind. I rely on plants for them. And I am really worried. When will they show up, and will these flowers be open? They're pretty darn tight, and they should be open in a couple of days. And I have observed a disparity. My zone, I'm in the far east part of my zip code. And it's different from the far east, because I was in the car yesterday driving around. Everything is spectacular, not where I am. In your county, Denise, you are in the middle, and it's distant north to south. And I just would invite some other observations when you have a big zip code where you have deviations throughout it, because it's troubling me. It's like a trip up. But I love journaling. I really do. And now I'm going to do it more seriously. And I have a project, the study of spring fruits, the flowers in prunus and malus in the rose family. Oh my God, I love those stamens, and the pistils seem to be fused. And I've always had a case where finding where stamens connect to the carpel. They're usually in a round series. So keep your eyes out, everyone. Keep your lenses handy. So I've just confessed my craziness.
Thank you so much, Jean. That was wonderful, folks. You can see why I asked Jean to give us a walkthrough. Her professional life and her journaling is really great. Jean.
I've named my journal. It's my ology journal, and it goes all the way down to psychology for me and sociology for everyone I can touch.
Thanks, Jean. So guys, we're going to do quick breakout rooms. And what I'd like you to do is share what you're noticing. That can be what you're noticing outside, or maybe what you're noticing about your new perspective or deepened perspective, maybe about phenology. So about 10 minutes, we're going to end there. You'll come right back into this room, add one thing that you've learned from today's session in the chat, and then we'll adjourn. So I'm going to go ahead and open us up. Thanks, folks. As you come back to the room, I want to end. I don't want to take too much of your time, but thanks so much for joining us. I hope you enjoyed the session as much as I did. A lot of great stories and great experiences. Great to see all of your faces. I hope you like the breakout rooms. Please offer something in the chat box. Give us something that stood out to you today, something you learned or something you'll remember. And I appreciate all of you being here.
Denise, thanks for doing such a great job and leading us into this journey.
Yeah,
you're awesome. Yeah. I had to say that.
I wish we were all together. It was,
but you know what?
Much more together. But
we got virtual love and we got virtual hugs. Okay, we can still do that. Okay, until we meet. Thank you so much.
Thanks, Joe. Thanks, everybody.